Hello everyone, welcome back to my channel. How is everybody doing? Lately I have been making some PowerPoint about microbiology, and this is also a good review for ATI TEAS exam. So some of the questions in ATI TEAS are with connection to microbiology, although it's rare that it happens, but in some cases it will come up. So it's better to be prepared than be sorry. So this topic is about chapter 8 and chapter 9, which is the microbial genetics for chapter 8 and biotechnology and DNA technology for chapter 9. So I got this, some of the information, I got them from Microbiology and Introduction by Tortora and Funky Case. Um, so that's it. And again, I would recommend this PowerPoint for those who are taking TASE exam or test of English academic skills, because somehow some of the questions are derived from microbiology topics. Okay, so the first term we have is genetics. Genetics is a study of what genes are, how they carry information, how information is expressed, and how genes are replicated. So just a reminder, I'm here to help you guys review and not to lecture or, give further information. So this is just a study review and help you pass with your exam, whether it's a class or it's an ATI TEAS exam, okay? Okay. The second term is gene. Gene is a segment of DNA that encodes a functional product, usually protein. So this term came up on my ATI TEAS exam. So I would remember this one. Gene is a segment of DNA that encodes a functional product, usually a protein. Okay, next is the genome. Genome are all of the genetic material in a cell. Genome is all of the all of the genetic material in a cell. While genotype is an organism's genetic makeup or allele combinations. Genotype is an organism's genetic makeup or allele combination. Genomics is the molecular study of genomes. Genomics is the molecular study of genomes. Phenotype is the expression of the genes. Phenotype is the expression of the genes. Watch out, because this might come in your TEAS exam too. So ATCG are the base pairing rules for DNA. ATCG are the base pairing rules for DNA. And for RNA, we have the uracil, ATUCG. That's for RNA. But we're talking about DNA in here, so you just only remember the ATCG base pairing rules. A stands for adenine, and T stands for thymine. C stands for cytosine, and G stands for guanine. Okay, just a reminder. Okay, substitution. Substitution is the most common type of mutation involving a single base at one point in the DNA sequence is replaced with a different base. Okay, again, substitution is the most common type of mutation involving a single base at one point in the DNA sequence, which is replaced with a different base. Okay, mutation is a permanent change in the base sequence of DNA. Mutation is a permanent change in the base sequence of DNA. Frameshift mutation. Frameshift mutation is one or a few nucleotide pairs are deleted or inserted in the DNA. So I can remember this one in my microbiology class. So when you think of frameshift mutation, think of it as nucleotide pairs are being deleted or inserted in the DNA. Spontaneous mutations are base mutations and our base substitutions, and frameshift mutations may occur spontaneously because of occasional mistakes during DNA replication. So spontaneous mutations is when base substitutions and frameshift mutations may occur spontaneously or at the same time because of occasional mistakes during DNA replication. Next, mutagens. Mutagens are agents in the environment that directly or indirectly bring about mutation. So mutagens, once again, is an agent or agents in the environment that directly or indirectly bring about mutation. Okay, chemical mutagens such as nitrous acid can convert the base adenine such that it pairs with cytosine instead of thymine. Other chemical mutagens are nucleoside analogs, benzopyrenes or benzopyrenes, smoke and root can cause frameshift mutations. Okay, for radiation, X-rays and gamma rays are potent mutagens because of their ability to ionize atoms and molecules. Then organisms have enzymes that can repair UV-induced damage, thymine-thymine dimers, but they can only repair so many, and if exposure is high, there will be dimers that are not repaired, then also known as a cancer. Then E. coli in recombinant DNA. Why is E. coli used in recombinant DNA work? It is used because it is easily grown and its genomics are known. So E. coli is a preferred host for gene cloning due to the high efficiency of introduction of DNA molecules into cells. Replica plating. Replica plating is the technique allowing identification of mutants. So replica plating, again, is a technique allowing identification of mutants. Bacteriophage. Bacteriophage is a virus that infects bacteria, also called a phage. Phage. Okay, transposons. Transposons are jumping genes, and they are small segments of DNA that can move or be transposed from one region of DNA molecule to another. So those are transposons. If you think of transposons, think of jumping genes or jumping jack. Just kidding. Okay, plasmid. Plasmid are a small ring of DNA that carries accessory genes separate from those of the bacterial chromosome. So again, plasmid is a small ring of DNA that carries accessory genes separate from those of the bacterial chromosome. Okay, next is conjugation. Conjugation in bacteria, the direct transfer of DNA between two cells that are temporarily joined. Conjugation in bacteria, the direct transfer of DNA between two cells that are temporarily joined. Okay, transformation. Transformation is a change in genotype and phenotype due to the assimilation of external DNA by a cell. Again, transformation is a change in genotype and phenotype due to the assimilation of external DNA by a cell. Transduction. DNA is transferred from a donor cell to a recipient via a bacteriophage. So during transduction, DNA is transferred from a donor cell to a recipient via a bacteriophage. Restriction enzymes. Restriction enzymes are enzymes that cut DNA at a specific sequence of nucleotides. So those are restriction enzymes. They cut DNA at a specific sequence of nucleotides. Then we have the ligase. Ligase joins Okazaki fragments in DNA. Gel electrophoresis. So gel electrophoresis is the process whereby an electrical field is used to move fragments through a porous substance similar to agar called agarose. Okay, so gel electrophoresis is a process whereby an electrical field is used to move fragments through a porous substance similar to agar called agarose. We have the recombinant DNA. Recombinant DNA, DNA that has been formed artificially by joining constituents from different organisms in vitro. So that's recombinant DNA. DNA that has been formed artificially by joining constituents from different organisms in vitro. We have the endosymbiotic theory. Endosymbiotic theory means that eukaryotic cells formed from a symbiosis among several different prokaryotic organisms. Again, endosymbiotic theory means that eukaryotic cells formed from a symbiosis among several different prokaryotic organisms. Next is the Burgess Manual. I'm looking for my husband to see if he's already there. So Burgess Manual is the most widely used source of information for the classification and identification of bacteria. So I remember during our microbiology class, if we don't know anything about the bacteria, we just go and refer it from the Burgess Manual. But most lab, they only provide one or two Burgess Manual, unless you have your laboratory is huge and a lot of resources are available. Okay, next we have the PCR, or polymerase chain reaction. PCR is a laboratory technique for amplifying DNA in vitro by incubating with special primers, DNA polymerase molecules, and nucleotides. So, where do we use PCR? I think when trying to detect DNA, right? Or like in forensic investigation. Okay, so PCR, polymerase chain reaction, is a laboratory technique for amplifying DNA in vitro by incubating with special primers, DNA polymerase molecules, and nucleotides. Then DNA fingerprinting. DNA fingerprinting is analysis of sections of DNA that have little or no known function but vary widely from one individual to another in order to identify individuals. So again, DNA fingerprinting is commonly used in forensic investigation. Analysis, which means it's the analysis of sections of DNA that have little or no known function but vary widely from one individual to another in order to identify individuals. Then we have the Krebs cycle, or the citric acid cycle. In Krebs cycle, you can see the cycle of reactions in mitochondria that begins with citric acid. It breaks down an acetyl group and produces CO2, ATP, NADH, and FADH2. That's for the Krebs cycle. Then we have the glycolysis, or glycolysis. I would read this as glycolysis. Glycolysis is the breakdown of glucose by enzymes releasing energy and pyruvic acid. So glycolysis, it involves breakdown of glucose by enzymes and then releasing energy and pyruvic acid. Okay, so this illustration is the mechanism of genetic transformation in bacteria. So I just put it in here because when I took microbiology class, our teacher would ask us to draw this during the exam. So it would be nice if you can memorize this one and if you can familiarize this mechanism of genetic transformation of bacteria. Okay, we have the recipient cell A, B, C, D, and it has a chromosomal DNA, and then DNA fragments from donor cells. So the step 1, recipient cells takes up donor DNA. Then step 2, donor DNA aligns with the complementary bases, which is the second tablet or capsule. Then the third picture, recombination occurs between donor DNA and the recipient DNA. Then it degraded and recombined DNA, and it will show you the genetically transformed cell. So that's it. Then the next slide is, this is the transduction by a bacteriophage. So in these steps, you have the first step, a phage infects the a donor bacterial cell. Then number 2, phage DNA and proteins are made and the bacterial chromosome is broken into pieces. And then number 3, occasionally during phage assembly, pieces of bacterial DNA are packaged in a phage capsid. Then the donor cell slices and releases phage particles containing bacterial DNA. Then number 4, a phage carrying bacterial DNA infects a new host cell, the recipient cell. Number 5, final step, recombination can occur, producing a recombinant cell with a genotype different from both the donor and recipient cell. So this is from the Pearson book, which is the Microbiology by Tortora and Funky case. So I just put it in here so that you can remember what it looks like. And I'm sure, I hope your teacher will not let you draw this one during the exam because it's kind of hard to draw it, but just try your best and good luck. Okay, so peptide bond. The peptide bond are the chemical bond that forms between the carboxyl group of one amino acid and the amino group of another amino acid. Again, the peptide bond is the chemical bond that forms between the carboxyl group of one amino acid and the amino group of another amino acid. So another term is autotroph. Autotroph is an organism that can form nutritional organic substances from simple inorganic substances such as carbon dioxide. So this, I just included this little picture. Again, an autotroph is an organism that can form nutritional organic substances from simple inorganic substances such as carbon dioxide. So this is the picture of a bacteriophage. So bacteriophage is a kind of virus that infects bacteria. It looks like a spider, huh? It has DNA and it has protein coat and it has a sheath and core and cell wall. Okay, and this one is a picture of carbon dioxide gas or carbon dioxide molecules. We have 2 oxygen atom and 1 carbon atom in the middle. Then ethanol, this is ethanol. The other, the symbol for ethanol is CH3CH2OH. Ethanol is a kind of alcohol present in beverages. Okay, if ethanol is your favorite, then high five. Just kidding. Okay, lactic acid. Lactic acid are produced in muscle cells from the reduction of pyruvate under anaerobic conditions or without absence of oxygen, that's anaerobic, to regenerate NAD+ so that glycolysis can continue. A rise in lactic acid usually accompanies an increase in physical activity. So if you have, if your lactic acid is so high, then it's because of physical activity like running and stuff like that. Okay, ligase. Ligase is an enzyme that connects 2 fragments of DNA to make a single fragment. So when I think of ligase, I think of a glue because of the term it connects 2 fragments of DNA to make a single fragment. Okay, mutagens. Mutagens are physical and chemical agents that interact with DNA to cause mutations. So some of the mutagens are radiation, chemicals, and infectious agents. For radiation, we have UV radiations and X-rays. And for chemicals, we have smoking cigarette and benzoyl peroxide, which is, found in, beauty products or in acne products. It's found in ingredients of acne products. And we have the infectious agents. We have the human papillomavirus or HPV and Helicobacter pylori or H. pylori. H. pylori or H. pylori. Okay, then we have the plasmid. Plasmid is a small ring of DNA that carries accessory genes separate from those of the bacterial chromosomes. When I think of plasmid, I think of a ring. It's round, so that's plasmid. As you can see, it's in blue picture, rounded. Okay, then we have the recombinant DNA. Recombinant DNA are DNA that has been formed artificially by combining constituents from different organisms. So here is the picture of recombinant plasmid, which is the colorful ring in the rainbow. And then we have here the E. coli, which is the host cell, and the transformed cell, which is combined recombinant plasmid and E. coli. Then we have the rRNA or ribosomal RNA. So ribosomal RNA is an organelle that contains most of the RNA in the cell, and that is responsible for ribosome function. So here is an illustration of our RNA function. Okay, here we have the transformation DNA. Transformation DNA is a change in genotype and phenotype due to the assimilation of external DNA by a cell. So this is a little bit easy, but when you actually draw it during the exam, it's kind of complicated, and you just have to familiarize the structure and then practice, practice, practice to make your work perfect. This is the genetically transformed cell. Okay, then our plasmid. Our plasmid is a bacterial plasmid that carries genes for enzymes that destroy particular antibiotics, thus making the bacterium resistant to the antibiotics. So our plasmid has the connection to resistancy to the antibiotics. Then we have the pentose phosphate pathway. Okay, I just put it in here, but you don't really have to know this unless you know you're gonna do it or gonna draw it in your exam. So pentose phosphate pathway are metabolic process that produces NADPH and ribose 5-phosphate for nucleotide synthesis. Okay, we have here the electron transport chain. So electron transport chain is a sequence of electron carrier molecules, membrane proteins that shuttle electrons during the redox reactions that releases energy used to make ATP. So this is how it looks like. It's a little bit complicated, so I'm not going to explain it to you. Okay? Just so you can have an idea as to what it is. I'm only doing the definition for electron transport chains. Okay, next we have the fermentation. Fermentation is a catabolic process that makes a limited amount of ATP from glucose without an electron transport chain, and that produces a characteristic end product such as ethyl alcohol or lactic acid. Again, fermentation is a catabolic process that make a limited amount of ATP from glucose without an electron transport chain, and that produces a characteristic end product such as ethyl alcohol or lactic acid. So here we have the illustration: 2 pyruvic acid, and it goes to 2 CO2, 2 NADH in the bottom, and 2 NAD plus equals 2 ethanol. Okay, then we have the glycolysis. Glycolysis is a metabolic process that breaks down carbohydrates and sugars through a series of reactions to either pyruvic acid or lactic acid and release energy for the body in the form of ATP. So glycolysis again is a metabolic process that break down carbohydrates and sugars through a series of reactions to either pyruvic acid or lactic acid and release energy for the body in the form of ATP. Okay. Then we have the Krebs cycle or the citric acid cycle. So a Krebs cycle is a cycle for releasing energy that takes place in the mitochondria. And in this process, first 5 carbon sugars and 1 carbon dioxide are taken from the air and combined. Then ATP then splits it into 2 groups of 3,3-PGA, and NADPH is given a hydrogen molecule creating G3P. 2 of the GSP groups are used, leaving the rest to be recycled by RUBP so that the cycle can restart. This is kind of complicated, so I don't think you're gonna have this in the exam, but it's just a good information. Then we have carbohydrates. Carbohydrates are a large group of organic compounds occurring in foods and living tissues, and including sugars, starch, and cellulose. Again, carbohydrates is a large group of organic compounds occurring in foods and living tissues, and including sugars, starch, and cellulose. Okay, and then we have the photophosphorylation. Photophosphorylation is the process of generating ATP from ADP and phosphate by means of a proton motive force generated by the thylakoid membrane of the chloroplast. chloroplast during the light reactions of photosynthesis. Again, photophosphorylation is the process of generating ATP from ADP and phosphate by means of a proton motive force generated by the thylakoid membrane of the chloroplast during the light reaction of photosynthesis. Then we have the carbon fixation. Carbon fixation is the initial incorporation of carbon from CO2, from carbon dioxide, into an organic compound by an autotrophic organism, such as a plant, another photosynthetic organism, or a chemo-autotrophic prokaryote. Okay, carbon fixation is the initial incorporation of carbon from CO2 into an organic compound by an autotrophic organism, such as a plant, another photosynthetic organism, or a chemo-autotrophic prokaryote. Then we have photosynthesis. Photosynthesis is the process by which plants and some other organisms use light energy to convert water and carbon dioxide into oxygen and high-energy carbohydrates, such as sugars and starches. Okay, again, photosynthesis is the process by which plants and some other organisms use light energy to convert water and carbon dioxide into oxygen and high-energy carbohydrates such as sugar and starches. Then we have heterotroph. Heterotroph is an organism deriving its nutritional requirements from complex organic substances. Again, heterotroph is an organism deriving its nutritional requirements from complex organic substances. And also, it's an organism that cannot make their own food. Heterotroph is an organism that cannot make their own food. Okay, we have the anabolism. Anabolism is the constructive metabolism, the process of building up larger molecules from smaller ones. So it's like, joining together. Constructive metabolism, the process of building up larger molecules from smaller ones. And then we have catabolism. Catabolism, on the other hand, is the metabolic pathways involving breakdown of complex molecules in living organisms to form simpler ones, together with the release of energy, destructive metabolism. So this is the opposite for anabolism, catabolism. Then we have the dipeptide bond. Dipeptide bond is the peptide bond linked by another two amino acids. So these are an example of dipeptide bond. So in here, there's two chemical compound that are joined together to form peptide bond. So here is in this picture, it says a molecule of water is removed from two glycine amino acids to form a peptide bond. Okay, so that's it, and thank you for watching my videos. And if you like this video, don't forget to subscribe and share. And this one is good for your study reference for your ATI TEAS exam, or if you're studying for your microbiology class and want to pass your exam. Have a nice day. This is Beth from Reality Life series saying bye.